Okay, fantastic. Thank you very much, Neil, for the introduction. Thank you so much for the invitation to Neil and Lorenzo. It's a fantastic privilege to be here today and to give you a lecture entitled to Rodolfo, who I have known for over 20 years now. In fact, a little anecdote for you. It took me a while to realize how long ago I was here, and there were no, I lost my email because I moved institutions so many times. There was no smartphones at the time, so there were no pictures and of embarrassing night dancing of me and my mates at the time. And I had to trace it back based on remembering going to a concert of a particular artist that would have only been in Italy once in a while. So that's how far back I was here. So I don't look that old, do I? So that was really the start for me. Coming to this course was the start of a career in epidemiology. I wasn't quite sure when I arrived because I had trained in physics before, and I was just temporary working at a hospital with a cancer registry, and I wasn't sure what the future held for me. But coming for 3 weeks to share the experience with fellow future epidemiologists from all over Europe, and being in the presence of eminent thinkers in epidemiology at the time and still now, really changed my mind and changed my life forever. So I hope that, no pressure on you guys, but I hope that you really make the most of the connections that you've made, both between yourselves, with your cohort, but also with the lecturers and the faculty. Okay, so this is the first time I give this talk. So you're a bit my guinea pigs today. You're my lab. And I really want to talk to you about data science and about how epidemiologists can successfully incorporate data science techniques, data science thinking, and the tools of data science into their epidemiological research. To contextualize, I'm going to refer to examples from work that I do in my own group, which is on maternal and child health. And as Neil said just now, I have the privilege of working at this new research institute, which is a flagship institute for research into the life sciences that sits just outside Milan. It was founded 4 years ago. Because it's a very new institute, just like a little word about what it does. The main unique selling point of the institute is to bridge across scales, which means that we have groups and centers that go from structural biology, really studying the structure of molecules, RNA, proteins, etc., through to tissue biology, cellular biology, and then studying organs, organisms, and us in Health Data Science Center, we study populations. The remit of my center is to, the long-term vision is to eventually improve the health of populations using the power of large-scale data and advanced analytics, which basically means data science. And I just have a picture here of my group, not incredibly updated, but just to remind myself and you guys that most of the work I'm going to share today is thanks to these really talented and excited young individuals. So what do I mean when I talk about big data? Well, my subtitle here is Big Data, from Big Data to Big Questions. Well, the type of big data that we use in epidemiology, which is both now desirable and inevitable to get hold of and to acquaint ourselves and to start using and harnessing, are in very different domains. But here I just have 3 examples of data that you've seen before and you've been taught about in your lectures. And they are mainly in the areas of electronic health records, which cover wide populations, so national-based registries and other databases of healthcare utilizations, so hospitalizations, drugs, etc. The second category is high-throughput molecular data. So in this case, the data is big, not just because it pertains to many, many individuals, but also because it's high-dimensional with respect to many different omics data being collected. For example, 7,000 proteins or 30 million single nucleotide polymorphisms when we talk about genetics. Environmental data is another source of really large data, both with high spatio-temporal resolution, so high dimensional in time and space. But I talked about data science without defining it, which is a mistake. So going to the Royal Statistical Society, the definition of a data scientist is that it's a job that uses tools and methods from statistics and machine learning to piece together relevant data, build models to create insight into big data for companies. I don't know what the for companies is there, but that's what they obviously pertains to a very applied position and applied skill set. So it reminds us that data scientists are applied scientists. They don't tend to develop new methods and new tools, which is what mathematicians and statisticians do. And they don't develop systems for storing data, managing data, which is what computer scientists do. But they really sit at this interface between domain expertise, which means the people that understand the data generating mechanisms and where the data come from and what the data mean, the mathematicians and statisticians that develop the tools, and the computer scientists. So data science is not new, and data scientists actually claim one of the most eminent and first data scientists was Florence Nightingale, although the statisticians say that Florence Nightingale was one of the first statisticians in modern history. So Florence Nightingale was a nurse, a British nurse that was working with soldiers from the Crimean War, and realized that most of the soldiers, anecdotally, seemed to die in hospital, not from their wounds, but from infections. Although this was an anecdote, and she realized that it was important that there was power in the data, in a systematic collection of the data, recording of the data, and analysis, and especially visualization of the data. So she set out to do just this, to become one of the first people to pioneer from data collection through to visualization, the whole pipeline of data science, and invented pie charts in the meantime. So today, of course, we live in a very different world. The landscape of data has changed completely, and if we think about data, and the first thing that comes to our mind is that we are totally inundated by data that already is there, or data that sits there, data that can be used, and we call it for secondary use, or a purpose is different from what they were originally collected for. So we have this luxury of not having to go out like Florence Nightingale did and collect data bed by bed, or like Jon Snow did, house by house, knocking around Soho in London during the cholera epidemic. So because of all this data delusion, because of the world that we live in today, and because of the skills that are required to be successful in an education today, are beyond, well beyond just regurgitating knowledge. There's a 21st century educational framework that is often talked about also in schools and involves these 4 different elements. This applies to all aspects of contemporary education, but I would argue this is particularly salient when it comes to epidemiology and the use of data science in epidemiology. So we'll go through this framework for the talk, starting with collaboration. So a data science approach allows us to collaborate in order to collect, collate, and connect data. I pause here because investing in a data infrastructure is your biggest asset and where everything starts from. You probably would have heard before that garbage in means garbage out, which means that unless your data systems and infrastructure is sound, the results that you're going to obtain are not going to be robust or reliable or replicable. So when we talk about the data infrastructure, there's several elements that we can think about. And the first one is whether the data is primary or secondary. By primary, I mean collected on purpose, for example, starting a cohort of individuals consented individually and with blood taken for biomarker analysis, etc. But this can also be augmented by existing secondary data, just like it's been done in the famous example of the UK Biobank study, where a cohort collected for primary purposes has been linked with existing data on their healthcare utilization with a long-term follow-up, so allowing retrospective follow-up and prospective follow-up at very limited cost. We talk about interdisciplinary data sources, so joining forces with colleagues that are domain experts in different areas, such as climatologists and weather experts, experts in geographic information systems, etc. Also experts in molecular biology when we go to the omics layers of information. And we talk about data that has to increasingly represent diverse populations, because unless we have underserved and underrepresented individuals and populations in our data systems, we can't possibly achieve health equity, far from it. Finally, data has to be harmonizable and adhere to the FAIR principles of findable, accessible, interoperable, and replicable. And here the picture I've chosen to illustrate the example is that of a pipeline that we've recently implemented in our center that takes data from genotypic and proteomic type of information and prepares summary statistics after quite a complicated, convoluted system of checking, including secondary information from publicly available databases, etc. But the idea is that the whole pipeline, the code behind it, is completely transparent, replicable, and open source. So I talked about big questions in the title, and really what we want to do is use the big data to answer important population health questions. So one question that really is so important to me, especially right now, that I don't think we can escape, is that of attribution of climate effects and climate impacts to population health. So a recent systematic review that was commissioned by the Wellcome Trust found that over the world, most of the climate impact studies concerned the region of Africa, some of them parts of Asia, and were limited, very limited in Europe, just to the effects, to narrowly studying the effects of smoke and fires. But really we are very far from building a picture whereby we know around the world over time how does climate and mitigations against climate change impact different domains of population health. And my particular focus is in maternal and child health, and we can see that the little child icon shows us that the only studies really come from the African region and a few from Southern Asia. So how can we improve on this picture? How can we build a more cohesive picture that takes data from everywhere? So it comes to no surprise to you that climate change, man-made climate change has resulted in a sharp increase in global average temperatures. And we know this because there's been models that really take the observations from around the world and put them together through supercomputers and create predictions that can be used at a really fine level along this grid for the whole Earth, so that we have achieve really high resolutions in time and space for temperature and also predictions of climate events. So why is this important? Because this can be linked with spatiotemporal resolution, with time and space data. It can be linked with existing electronic health records. There are secondary data can be used for covering entire nations. This is incredibly time-consuming work and requires a deep and long investment and collaboration to bring together all these different systems that don't talk to each other and are not necessarily harmonizable at face value. And recently I was part of this large consortium that was funded by the Wellcome Trust for weather attribution and climate attribution impact that achieved pooling, well, hopefully will achieve a pooling of 45 million mother-child pairs collected from, with information collected from in silico cohorts, which means from electronic health records linking maternal and child records. At the moment, most of the data comes from Western countries and Africa. We really need to move towards making it more equitable and including more diverse representation. So what do we do with this data? This is an example from another study that is a more limited but more finer modeling type of study that includes data from Italy, the Lombardy region around Milan, where we are around there, the UK, in particular in England and Wales, and Sweden nationwide, and pooling data from these 3 different regions in Europe. We are trying to investigate both individual level vulnerability of, in particular, perinatal health and preterm birth to changing temperatures. So by groups of women, are there characteristics that mean that they are at increased risk in like a fat modification type of framework, but also at the population level, we're looking at mitigation measures, and we're looking at whether there's any suggestion that climate change adaptations have been working. The second element that I want to talk to you about today is that of creativity. So by creativity, I mean a way in which we can describe, visualize, and detect patterns, so hypothesis generating work. Epidemiologists have been using descriptive studies for a very long time, and there were some of the first study designs that were used that give them rise to hypotheses to test in etiological studies. But perhaps we can find other data types and other ways of using these data types and visualizing data types that make us things that we can use the things that jump up to us, patterns jump up to us, and we can detect important changes worth investigating. So the creativity comes not just in the description and the high dimensionality of the data that we can see through data science, but also through the type of visualizations. And I think creativity also goes into the process of creating what's called synthetic data. This was, I think, a very smart and creative way of going around the problem of sharing personal data and disclosing potentially sensitive information. And synthetic data means a way of simulating the type of data that you would want to operate on, you want to create insights from, so that you can train your code on this simulated data and then not really ever disclose the rows and columns of the actual cells of real data. So really with data science, in a creative and data description way, we can think about new ways of tackling old problems. So the big question here is, why do preterm birth rates vary so much across the world? So preterm birth, pretty straightforward to study, defined mostly by gestational age at birth, and a child is preterm if born before 37 weeks of gestation. We see that worldwide the prevalence varies dramatically. In Italy it's about 5%, and in Bangladesh it's almost 20%. So how could it be that we have geographic variations that are almost fourfold? So we, at the moment, despite this being one of the most studied perinatal outcomes, we're still lacking a satisfying description of the determinants of preterm birth. So there's still room for improvement in the detection of new, actionable, modifiable factors that prevent preterm birth. So what we thought about doing was starting from data from a healthcare utilization database from the region where our research institute is, that we have access to. So we have data for the whole of the region of Lombardy. Milan is around here. This is a really large region that covers a population of 10 million people. So for the Danes and the Scandinavians, it's like a small country. For this region, we have data covering just over 12 years of all the births that happen in hospital, which is the vast majority of them. And so we can track what happens in space and time to preterm birth and create descriptive trends. Now, this is not enough because by eyeballing trends, you can't see, oh, something definitely happened there, and look, now preterm birth is going up or it's going down. There's a lot of noise in the data, and you're not exactly sure what to base this trend change hypothesis on. So my postdoc had the idea of going to a database that exists that collects all the policies and the regulations that are relevant to the region. So this means a database of 23,000 regulations and policies that come into place with a date between 2012 and 2023. They are either European, Italian, or regional. For each of the policy, we can assign a target intervention. So is the policy targeting an environmental exposure? Is it air pollution, air traffic? Is it something to do with airports? Is it something to do with noise or with water quality? And we can identify a date where the implementation of the policy happened. So by text mining an existing large database of largely PDF files and convoluted legalese Italian text, we can hopefully find a way of identifying new natural experiments systematically. So the work involves assigning environmental exposures to each of the policy in the database, identifying the relevant date for each of the policy, perhaps more than one date with sensitivity analysis, a date of implementation and a date of when actually most of the region was actually effectively doing the policy, and analyzing trend changes in relation to this date of implementation, and so supposed change in environmental exposure, and then identifying which one of them out of this large number shows statistically significant or statistical evidence for a difference in the trend. So this is a series of natural experiments, a bit like if you had a genome-wide association study, you do a genome-wide, we do it policy-wide for a number of different policies. We don't know how many policies we're going to find that are definitely linked to environmental factors, and work is in progress to identify the distribution of environmental factors addressed by the policies. But the idea is this, to find the trends and then to, jumped up. And then to identify an environmental exposure that show promising association to then take forward into investigations of more causal investigations. So this is my creativity example. The next example is about critical thinking. So why did I say that after we've run perhaps 100 or 200 pseudo-natural experiments with the policy interventions, we then have hypotheses to take forward into more investigation? Well, I say that because the original idea wasn't to test a hypothesis, it was to describe and test across many different potential factors. And so it was to identify interesting hypotheses. So we need to be really critical about the results that are in front of us and think about how they were generated and what assumptions were necessary in order to run the models and generate those assumptions. So the critical thinking part, I think data science can help us with, is in evaluating alternative explanations behind the results that we find, especially in association studies. So this means investigating heterogeneity and different contexts. When a study is attempted to be replicated and we find different results across different countries, for example, or across time, so an effect modification, immediately we think, okay, the result wasn't real. But perhaps it means that this is unveiling an effect modification that we weren't thinking about, we weren't testing it for. And so heterogeneity is telling us that there is something else going on, sometimes really important. We need to consider assumptions and their potential violations for each individual model that we run, be it a linear regression or be it a t-test. Think about data generating mechanisms for each of the assumptions, and some of them are in the data generating stage. So how do we define variables, for example? How do we define disease? then perhaps if we use a certain cutoff, a sensitivity analysis should be run using a different cutoff to test how sensitive our results are to that choice of cutoff. So sensitivity analysis is one of our strongest weapons in the fight against just believing results and moving on, not using your brain. So let's not ChatGPT our whole epidemiological practice. I think one of the strongest ways in which we can be very critical about results is the deliberate use of positive and negative controls. This is something that lab scientists, basic scientists do all the time. Before they believe their results, they run positive controls. Not so much negative controls, but it does happen, for example, to calibrate apparatus. And it's only been recently, I think, in epidemiology that we've been making more deliberate use of this. And those of you who've been sitting in Deborah and Carolina's sessions over the course of the week would have seen quite a few examples. Again, we don't stop at face value, but we try to rerun not just the same model, but with slightly different variations with sensitivity analysis, not just replicating in different contexts, but we try to bring everything together. So we try to triangulate results from different angles using different data, using different model and modeling assumptions, and so that really we can be confident that the results are, if they all point in the same direction, that's the idea of the triangle, if they all point in the same direction, they're less likely to be caused by violations of assumptions or bias. So the big question here is, can we disentangle the association between stress, maternal stress, and breastfeeding? So I've been interested in breastfeeding for a while, and there's a huge amount of observational epidemiological literature that shows us that mothers who are stressed, who have undergone trauma, who are anxious, many different domains of maternal health impacted by stress, seem to have more problems with breastfeeding, and these are associations that show associations with stopping to breastfeeding early. So what can we do? Even adjusting for all confounders, we still find that it looks like maternal stress is associated with breastfeeding. We shouldn't really stop there. I think we should think about, for example, the timeline. We should think about when is maternal stress more important in determining perhaps premature cessation of breastfeeding. What can we do to help these women? But also we know often that these studies are retrospective, and we know that stopping to breastfeed early before you're ready to do can be associated, leads to postnatal depression, and also is very stressful. So, can somebody caption this picture for me? Maybe you've seen it before. So this is to remind myself that A, it's been 20 minutes already. B, that correlation is not causation. So what can we do? We can perhaps think about a causal inference framework that uses instrumental variables. I know in Irene's sessions, you've been going through instrumental variables, and maybe quite a few of you by now have seen this type of statistical approach. The idea of instrumental variables is that if we are interested in how an exposure, in this case maternal stress, causes breastfeeding cessation, but we're worried about the existence of a number of confounders that we can't measure and account for all in our study, then we take a proxy for the maternal stress. And the assumptions are that this proxy is not linked in any way with confounding factors, is definitely a strong predictor of maternal stress, and is not linked in any way directly with breastfeeding cessation other than via influencing maternal stress. So this is if we then choose an instrumental variable which is of genetic origin, a genetic variation, then this is a Mendelian randomization study. So the great thing about Mendelian randomization studies today, we'll talk about limitations later, is that there are huge amounts of publicly available high-dimensional data already collected from over 6,000 GWAS publications and 67,000 data sets of full summary statistics relative to different traits and different populations that tell us about potential genetic instruments for traits that we're interested in. So can we find some genetic predictors of maternal stress and maternal anxiety? Yes, we can. The Psychiatry Genetic Consortium has published on this GWAS on the left-hand side here for you. So what this shows is a Manhattan plot, and it shows dots that correspond to p-values of running different regression models. So this is not a plot of individual observations, it's a plot that summarizes results from 30 million regression models. And the p-values are plotted on the inverse log scale, so the higher the peaks, the higher the evidence against for that particular locus, that position in the genome to be associated with our exposure of interest. So here it looks like there's a few promising regions in the genome that we can use as instruments for predicting lifetime anxiety disorders. I've just plotted one here, but there's many other ones also for depression, post-traumatic stress disorder, stress reactivity, cortisol levels, etc. They're all proxies for maternal stress. And on the right-hand side, we have a new genome-wide association study, so a preliminary figure from a meta-analysis of genome-wide association studies on breastfeeding that my group has been working on in collaboration with Bristol and other colleagues. So whereas this GWAS is based on 250,000 people, it's a collaboration that has taken several years to achieve this sample size. Our GWAS is still stuck at 30,000, but we're hoping to improve some sample size soon, which hopefully will allow some of these regions to come through the threshold of multiple testing and show us some peaks on this side too. But we can start by investigating what we have so far. And the idea is, do we find some genetic overlap between the regions that predict anxiety and the regions that predict breastfeeding? So this is we start with the genetic correlations for GWAS is on all of these different traits for maternal anxiety, proxies for maternal anxiety, and find genetic correlations with breastfeeding. And we find that panic disorder for now, depressive disorder, and attention deficit hyperactivity disorder seem to associate genetically with breastfeeding. So all we know for now is that there seem to be loci that underscore both traits. We don't know about the direction of the association. We don't know if it's a breastfeeding or things underlying breastfeeding inability that will then lead to anxiety or the other way around. And it's important to go on in our investigation and be critical about this because, for example, the first locus that was found when the GWAS of lung cancer was run the first time was a locus for nicotine dependence. So it wasn't lung cancer that was causing nicotine dependence, it was nicotine dependence that was causing lung cancer. So genetic correlation between two traits doesn't mean necessarily that you already know what the direction of causality is going to be. So we tested this through what's called the two samples Mendelian randomization, which I'm not going to go into details about the methods here. But the idea was that we used, in particular, a GWAS that summarized both depressive disorders and anxiety together, so ever having had depression and anxiety. And each one of the dots here represent running a separate model that gives us the best causal estimate for the association between anxiety and depression in the mother and breastfeeding later on. So because we achieve lots of different potential estimates, we need to find a way of combining them together to find, is there a middle ground, do they agree with each other, just like you would do in a meta-analysis after a systematic review. And we do that through different models that give different weight to these different estimates. So some of them are not compatible with other ones, some of them are seem outlier, like the one down here. So the different lines of different color are built in within the package that already gives you 5 different weighted averages of the Mendelian randomization estimates. So already we can do an internal triangulation of our result, because if all the assumptions of all of those different models are met, then the line should converge. all of the lines should be exactly on top of each other, and you should have one reliable summary estimate of your Mendelian randomization experiment. But here, we're not in that situation. We have different diverging lines, and so for now, we will need to certainly go further with more sensitivity analysis and see why the lines are not overlapping each other, which ones we trust the most, how can we understand enough in order to choose one of those estimates, going from no effect whatsoever to a small negative effect. So I said we're really quite at the beginning of this exploration, and the next step that we want to do to keep being critical about this type of work is to go into deeper biological measurements and incorporate omics data into what we're doing. So one of the sources of omics data that is available to quite a few cohort studies now is proteomics. We have a data set of over 7,000 proteins measured in plasma and 10,000 individuals, 5,000 men and 5,000 women from a Bangladeshi study. We thought about using this to look for associations with breastfeeding and also with anxiety. But before doing that, because it was a fishing expedition to start with, we wanted to test how much we could trust the methods that we were developing to track these proteins and protein trajectories over time. And so we used a positive control. So our positive control was to think about a reproductive stage that we knew about the women in this cohort were going through, which was the menopause, and look for proteins in which we could find major differences between men in blue and women in red in their protein trajectories. And by using this positive control, we have now established a methodology that picks up the most relevant menopause significant proteins for this cohort. Now, having honed the methods and the pipeline, we can take this to then studying breastfeeding, for which we have no idea what proteins in plasma would be related to breastfeeding. So we started by narrowing down from the 7,000 proteins, narrowing down only here on 39 proteins, which we know from the Human Protein Atlas are associated with sex differences. So they are found in exponentially higher levels in women compared to men. And across these 39 proteins, for the 5,000 women, we find that for now, one protein shows an association with breastfeeding. But this, funnily enough, it's a pregnancy-associated protein, so perhaps it's a real finding. But these will need its own triangulation sensitivity analysis and taking it further to be as critical as we can with ourselves before we believe the results. So one thing the next one is communication. So I really wanted to talk to you about communication, and I had a few slides also about scientific publishing, which I then swiftly removed because that's what your next lecture is going to be about. But what I mean by communication in this framework is communication so that we can achieve our ultimate goal in epidemiology, which is that of implementing public health strategies and really improving public health. So a data science approach can help us, I think, in conveying balanced, evidence-based messaging. When I talk about balance here, I was really thinking about showing all sides of the coin. So showing uncertainty in our results and communicating uncertainty, showing potential unintended consequences of, for example, an intervention, vaccine side effects, not hiding any of these results, but also setting out to investigating unintended consequences. More and more we find efficacy studies that look at policy evaluations, but they forget to look at other aspects of population health and individual health that could be affected by the introduction of the policy. And I think that would be a really critical message to show our willingness more and more to test and be critical of potential unintended consequences. The second aspect that I think data science is really great at doing is effective messaging. Now, communication is absolutely important internally within a group, externally with your collaborators, but also with policymakers, after we conclude that we believe the results and we have something that we want to take forward to communicate. Infographics are really quite the next level of communication, really, because they're a visual way, and they're so much more effective, so much so that nowadays most journals require a graphical abstract. A graphical abstract is an infographic. And lastly, I wanted to talk quickly about fighting misinformation. So part of our duty, I think, and our role as communicators, using data, so attaining to what the data says and not so much our opinions, is that of being out there in the world and reminding the world of what the data shows. An informed interpretation of the data, a critical interpretation of the data, but what the data show. And it's so important, I think, to fight misinformation that we as experts, as domain experts, don't shy away from occupying those spaces, because if it's not us, somebody else will use the platforms and somebody else will get the message out. So when you press release a paper, when people ask you for your opinion, to give interviews, to write about it, talk about it with the public, public engagement events, don't shy away from doing it. It's a very important part of your work, and data science can help you with visualizations. And the whole process really is important in getting to the end and not retreating in our ivory towers and getting our metrics because we've published a scientific paper. So I think I've gone too quick. More time for questions. So in conclusion, a data science approach for epidemiology, I think, is one that is both desirable and inevitable because the data is there. And again, if we don't use it, somebody else will. We might just as well use it and use our competencies to extract the best information from it. Data scientists, remember, are not technicians, so they're not there just to create pipelines and to manage your data, but they are trained in getting insights from the data, and in asking questions, and in being curious about the data, perhaps with different intrinsic biases, for example, training biases from the ones that we might bring. So a collaboration, a really deep connection and collaboration with data science, I think, with epidemiologists and our expertise in finding the most relevant public health questions and clinical questions for the matter for individuals and population is the way to go. And I really think that working together, we can harness big data to answer the big questions. That's it. I wanted to thank, false finish. I wanted to thank my team of incredibly enthusiastic individuals. My team is last night I was talking to some of you about the lack of epidemiologists in my team, and I saw that as a problem, but actually it's a major asset to the work that we do, because we have the luck to have molecular biologists, statistical geneticists, data scientists, and statisticians that see problems from different angles and really help me conduct this work. What it means is that it requires a continuous communication between us to make sure that we use the same language and that we create a shared language and an understanding of where we're going with an epidemiological framework, but also how we're getting there. We talk about the same things. So it's very stimulating, and there's a two-way interaction always, so it's very refreshing. The whole Health Data Science Center at Human Technopole for funding me and supporting me and giving me the opportunity to carry out this work. The many collaborations both in Italy and elsewhere, and I can't forget to thank enough the University of Bristol for training me, I don't get emotional, for 18 years and really allowed me to become the epidemiologist I am today, and the other collaborators that contributed to some of the work that I showed you today, and the funders. Thank you.